First needs to be legal response. Sorry.
Good morning, everybody.
Sure, I would. I don't know if I am.
We'll get started with grand rounds. Thank you all for being here. I'm grateful for the morning. It's all good. No, it's absolutely fine. Thank you for the amazing attendance that we've had for the last three grand rounds. I really appreciate that and the engagement. So great work. I'm really honored today to be able to introduce Dr. Danny Rowe, who's our grand round speaker. I thought that the timing would be perfect to start with something that's fundamental for surgeons, which is wound healing and the biologic basis for that, and maybe a new twist on it. Dr. Rowe is currently assistant professor of surgery at the Boston University School of Medicine, where he serves as vice chair for research in the department. Dr. Rowe had completed his undergraduate education at Duke University and then underwent MD PhD training at the University of Pittsburgh, where he was a corneal biologist. And then he saw the light, no pun intended, and decided to do a plastic surgery residency at Harvard, the combined Harvard program, and since then has now joined the Boston University. He's a prolific surgeon scientist who holds 2 NIH grants, and in addition to that, has other external funding, and really is dedicated to understanding the effect of senescence, that is aging at the cellular level, on the biology of wound healing. And so I'm really excited for this lecture and grateful that you would come down and educate us.
Yeah.
Thank you so much.
He wasn't here.
Okay, we'll start with the delay here. Let me just get my pointer going here at least for you guys. Okay, well, thank you so much for the invitation to be with you today. Today I want to share some of the areas of research that I kind of have a passion for, and that's understanding aging, the biology of aging, as well as care of surgical patients. And I think hopefully today you can come away with some ideas of how we can approach our older patients as they undergo more surgeries in the future. So I want to start by kind of addressing like why I'm here. Maybe asking like who invited this guy for grand rounds. And I've known Laura Niedenhofer now for I think at least 18 years is what I calculated actually. She's the current director of the Masonic Institute of Biology of Aging and Metabolism. And she's been a great mentor of mine over the last now almost 2 decades, and really has inspired me to kind of pursue this type of work that I'm doing now. So that's probably one of the big reasons why you're kind of stuck with me this morning. And the other part I wanted to acknowledge is Dr. Ikamunin, who really is pioneering this bridge between surgery and aging, really at University of Minnesota, and has been a key pivotal influence in establishing this Minnesota Tissue Mapping Center for Cellular Senescence, part of this NIH SENDNet initiative. And it's really rare, I think, for surgeons to be kind of involved at this level in terms of aging research. And I think it's super important as there's no question that our patient population in general could benefit from this type of work. So I have 4 objectives for you today, is really describe the core principles of aging biology and neuroscience relative to surgical care. I'm going to take a deeper dive into senescence and wound healing and show you how aging actually corrupts this response. Provide some evidence that wound stress actually can drive systemic biological and functional aging. And then identify some emerging neuroscience-based diagnostic and therapeutic strategies that we could potentially think about to improve surgical outcomes for patients. So there's no question that we're getting older. It's a remarkable time right now, and especially in developed countries in which the life expectancy is now surpassing 80 in many developed countries. But with that expansion of lifespan also comes issues with patients that are in medical, coming for medical care. This includes more complex patients that we're seeing in terms of accumulation of disease and biological aging that we're now beginning to measure. So despite this increase in life expectancy, really the intersection of health span and lifespan is becoming kind of a new focus. And this is where kind of neuroscience comes into play, and I'll explain more what that means in the next slide. I follow a ton of research blogs and things on aging research. This is one that really captured my attention more recently, and you may have come across this in the news as well, because it really became quite the headline. And it was a study out of Stanford University in which they found they traced about 100 or more folks and did extensive multi-omic profiling to establish that aging is not really this linear trajectory. You actually have areas where you have these incredible spikes of aging. And they found, using this multi-omic analysis, that there's these huge spikes around age 44 and age 60. And whether or not this actually plays out in the future is to be determined. But I think the principles here are that we can now measure aging at a whole nother level that we've never had before to determine what's driving some of these processes. And as someone that kind of recently turned 45, I think in retrospect, I can definitely sense that these spikes are happening. And I'm sure there's some folks in the room who are also kind of experiencing these really peaks in aging that happens. So it's an exciting time also now because we're beginning to find out that aging is there's mechanisms behind things that we haven't discovered before, and that these mechanisms are potentially modifiable. So now, before this concept of longevity science and neuroscience was kind of a little bit more on the fringes of biology, but now I think it's certainly becoming a lot more mainstream. And one of the most kind of public examples of this, and this is to the far extreme of what's happening, is the story about Brian Johnson, who many of you probably have heard about in the public. He's this tech CEO, venture capitalist, millionaire, who really started this anti-aging campaign for himself to the absolute extremes. It's reported he spends almost $2 million a year in order to undergo various protocols for sleep, exercise, etc., and all these other advanced therapies that he's trying to actually improve his biological age. And his motto is "Don't die." And he thinks that he can achieve potential towards immortality with this. And on the right of the screen, you can actually see some evidence that some of this is working in terms of the measurements that we're taking. And the point here is not that these are things that we should all be doing, but the point is that we can now measure a lot of these aspects of aging that we really haven't focused on before. And one of the key things are these DNA biological age markers, or these epigenetic tests that we can actually do to actually figure out what your actual biological age is versus your chronological age. And this kind of story in the middle of the screen is quite interesting. It was actually from a couple of years ago where he did this experiment where he transferred the plasma of his younger son, which is actually shown here as a 17-year-old, to himself. And he transferred his own plasma to his 70-year-old father. These concepts of plasma transfers actually have kind of played out in the preclinical space, to be honest. But the interesting anecdotal story behind this is that he felt there was no benefit from his 17-year-old son transferring to himself because he was so optimized already, so he wasn't seeing the effect. But when he transferred to his 70-year-old father, they report that he actually had a 25-year decrease in his epigenetic aging from that transfer. So something but not to say that we should do this, but it's kind of lays the foundation of how this is becoming kind of in the public. Now, I looked at this more recent review in 2024 of some clinical trials that are exploring these anti-aging mechanisms. And I kind of built this chart, and I'm not going to go through this in detail, but the concept here is that there are a lot of these interventions that are being tested in human clinical trials now, and there's a lot of biological basis for what they could potentially do in targeting aging. Things like metformin, low-dose rapamycin, the GLP-1 agonist is a very interesting story now. Dasatinib, quercetinib, fisetin, arsenilytics, which I'll talk a little bit more later. And the goal is, I think, as a surgical community, we should be attuned to what is happening in this space, and always think about how we can apply this to surgery. Not to say that we're going to all of a sudden start using these in surgical practice, but we should be aware of some of these potentially big changes in the aging research. And on the right of the screen, I'm sharing with you a kind of a summary of a analysis from Clarivate, which is this big life science and healthcare analytical firm. And they recently put out this analysis that is suggesting that this idea of longevity science is going to become the next big thing in investment. And they listed kind of these 10 different aspects that are really driving this forward, and they're predicting there's going to be massive investment in this space. And I think one of the big signs for this is actually pharma. It's actually getting into this space and creating aging-related divisions within their own pharmaceutical industry, which is really pushing this field forward. So let's take a step back and let's talk about what geroscience is and why should surgeons care about this. So it asks this single powerful question: why does aging drive so much disease? And the central hypothesis behind this, which was coined by Felipe Sierra, one of the directors of aging biology at the NIA, and considered the father of geroscience, is that aging itself, not any single disease, is the primary risk factor for most of the chronic conditions we treat. So rather than treating each condition separately, like hypertension, COPD, etc., you actually can target the biological mechanism of aging. You can actually treat many diseases associated with it. So, we all practice this kind of subconsciously or consciously when we see an 80-year-old patient and wonder why this patient's definitely not going to heal like a 20-year-old or a 40-year-old, or when we see a patient that seems a lot older or younger than their actual chronological age. So we're kind of thinking about this already. And what geroscience does is gives us the biological framework to move beyond just saying this patient is old towards kind of this patient has actually targetable aging mechanisms. And like I mentioned, as our population continues to grow significantly, it's actually estimated right now that 1 in 3 surgical patients are actually 65 years of age older, and in the U.S. alone, and then about 5 million of these older adults undergo surgery per year. This is going to double for the next couple of decades. And the other problem is when surgeries go wrong, they go catastrophically wrong for older patients, right? We have approximately 10.5 million Medicare beneficiaries that are affected by a wound or wound complication. when there is a complication, older adults with surgery like dehiscence or surgical site infections, you're talking about expanded mortality, morbidity, hospitalization, and cost. And, the chronic wound problem in the United States is a $22.5 billion expenditure, and most of these chronic wounds are really focused in the older population. So really, we should be thinking about, how can we really treat this growing population and achieve faster and more resilient healing. So I want to take a step back and introduce this concept of the hallmarks of aging, which was introduced by Dr. Carlos Lopez-Oten back in 2013. He was a molecular biologist that's in aging research. Originally, it was 9 hallmarks. He expanded it to 12, which I'm showing you in the pictures here. And I think actually even more recently expanded to 14. And these are hallmarks that are organized into basically 3 tiers. If you think about primary or the initiating damage that's happening, such as DNA damage, epigenetic alterations, loss of proteases, etc. And there's these antagonistic roles, which are the stress responses that are happening, like deregulated nutrient sensing, mitochondrial dysfunction, and cellular senescence. And then what effects this actually have on the tissues, stem cell exhaustion, inflammation, extracellular matrix changes, etc. And for me and a lot of others in the field, we think that cellular senescence is one of these hallmarks of aging that actually intersects with so many other of these hallmarks that if you can target this cellular senescence, that you might have the ability to target multiple aspects of the hallmarks of aging. So I'm going to take a deeper dive into senescence, and we have some of the world's most expert senescence in the field here right now, Lauren. But I'm going to introduce this concept to you because the rest of the subsequent talk is going to focus on senescence. So it's a moving target. I'm just going to preface it with that. But in my definition right now, it's a fundamental stress response that leads to a stable and irreversible cell cycle arrest, a general state of apoptosis resistance, and an active secretome. I apologize that this is small on the screen. So there are many causes of senescence, from DNA damage to radiation, and tissue injury is also certainly a feature of senescence. A cause of senescence. There are many features that come up in senescent cells, like dysfunctional mitochondria, protein modifications, etc. But there are no kind of universal features that you could say define a specific senescent cell. There's many different varieties that can happen. For the sake of today, we're going to talk about some of the more common markers that the field has used, and this is ever evolving. But these include P16, P21, which are cell cycle inhibitors, senescence-associated beta-galactosidase, SABeta-gal, which is a kind of lysosomal marker of senescent cells. But the real effector part of this is the SAS, or what's called the senescence-associated secretory phenotype. And you can see it's kind of a loose collection of a lot of these signaling molecules, interleukins, chemokines, inflammatory factors, etc., that really are secreted by these cells that drive its impact. And this can be felt in the surrounding tissues and organisms as impaired regenerative capacity, changing the behavior of the neighboring cells, modifying the ECM, losing function, and promoting persistent inflammation. And why should we care as surgeons? So it turns out that senescence can increase with aging across almost all the tissues in the body. It definitely increases with age-related diseases, has a strong association with that. And it can impair tissue repair, homeostasis. To the right, you just see a kind of a cartoon of various diseases that have been shown to have elevated levels of senescence associated with that disease process. And this was, just 5 or 6 years ago, and it's certainly expanded since that particular time. So the benefits of removing senescent cells has come from a really rigorous and abundant amount of preclinical data that have shown that if you target these cells and can remove them, you can actually prevent and treat age-related diseases. You can improve physical function and lifespan in old age. Many of these studies are coming from Lauren and Paul here actually themselves. The data has become so convincing that we're now trying this actually in humans in clinical trials. The last I counted, there's probably between 40, maybe more than 50 now, active senolytic clinical trials worldwide. So this is being tested in patients. And it spawned off at least 30 or more senolytic biotech companies that are focused on this. It's really quite a boom in what we're doing with senescence. But we got to pump the brakes just a little bit, because it turns out also that not all of this senescence is detrimental. And it's about kind of duration and persistence is really what it distinguishes these things. because we can see evidence of waves of senescence in development, in tissue regeneration in organisms such as salamanders and zebrafish that can regenerate limbs. You actually see this wave of senescence that comes in and goes away. If it doesn't go away, if you block it, it can't regenerate. So it's this idea that if senescence becomes more persistent and chronic, it's when you start to have issues and you have tissue generation or chronic inflammation that can drive age-related diseases. And again, it's not this binary phenotype either. There's definitely gradations between these two. But for the purpose of today, we're going to try to focus on kind of transient versus persistent. This is one of the kind of seminal papers, I think, in the field now more than a decade ago. It came out from the lab of Judy Campisi, who was one of the founders of this kind of senescence field. And back then, she created this really sophisticated mouse model called the P16-3MR transgenic mouse. So what this does, it's a transgene in these mice that whenever P16 is expressed, it has the P16 promoter here, it actually drives expression of this really cool protein that has a luciferase, which means it lights up cells, a red fluorescent protein, so you can theoretically trace them, and an HSV thymidine kinase, which means that you treat these cells with gain cyclovir, they'll actually go away. So it's a really cool model. And what they showed, and when they did wounding in young mice, it's a single wound on the back of a mouse in this picture here, you could actually trace these cells, and you saw that they started to light up on day 3, and they really lit up on day 6, and then it went away. So kind of perpetuating this transient nature of senescence. And what was cool, like because they could actually remove these cells with the gain cyclovir treatment, they could remove this response. And when they did that, they found that there was actually delays in healing that were happening. So they thought these cells were beneficial or essential for the acute wound healing process. And what was also interesting is that these cells were really just 2 to 5% of the total population, which means very few, very rare, but having a large impact. I think that's important because now my lab is really interested in the cell biology behind what these senescent wound cells are, what are their functions, and importantly, how does it change with aging? Where I mentioned we have these this accumulation of senescence that seems to happen. So in order to answer this question, we have young and aged mice, which is the equivalent of probably an 18-year-old or a 75-year-old human. Our model in our lab uses a single full-thickness dorsal wound. We don't do splints. We allow these mice to kind of heal very naturally. And we always find consistent delays in wound healing in our aged mice that take about 24 days to fully kind of heal, whereas most of our young mice are healing by day 18. So almost a week, 6 to 8 days of differences in healing rates. What was surprising to us, we found, this is looking at P16, again, that cell cycle inhibitor I mentioned before, expression in the tissues throughout this course. We found that there was a slight increase of P16 at baseline, which others have reported in aging skin. And we kind of found that same peak of expression of P16 that I just mentioned in that paper before in the young mice. But what surprised to us is that we actually thought we found a blunted response in aging, which we were expecting to see this just massive response. We looked at a couple other factors that are associated with senescence, including some of the SAS factors, and these were all really flat, which really caught us off guard, that aging actually has a diminished acute wound senescence response. So in order to kind of delve further into this question of what is happening, we use a technology called single-cell RNA sequencing, which I think which is great, everyone knows about now these days in the public even. We isolated the peak of wound senescence, which is day 6 in that young mouse, which I just showed you, and also the aged mice, and we're able to sequence every single cell basically in these wound environments, and just showing you what's called a UMAP plot below of the actual single cells. Each dot represents a single cell that we actually were able to sequence their active transcriptome. And we found the common players that are involved in wound healing. When we took a deeper dive to understand what the functions of these cells were, we had to come up with a definition of what we're thinking senescence was, which we decided to use P16 positive, P21 positive, K67 negative, which is a marker of proliferation. So really deep into the senescence phenotype. And we found that most of these cells, we found about 3% kind of consistent with what before, were actually wound fibroblasts based on their gene expression profile. And we compared all the gene expression to these data sets that exist already, looking at how senescent are they essentially. They did match with a few of the senescence gene sets that are out there, but not all of them. I think that's an important distinguishing feature because this is kind of another version of senescence, another flavor of it that has its own unique profile. And we looked at, kind of deeper into some of these themes that were coming up. We actually found things like development, morphogenesis, ECM, collagen, things that weren't typically expected, you would say, from a classic senescent detrimental cell. And when we look at gene expression for various extracellular matrix genes, collagens, et cetera, we found that this group of fibroblasts certainly had more or higher expression of these ECM-related proteins, which was surprising to us. And this doesn't just happen in the mouse. So we actually analyzed publicly available data from a human experiment that was published recently. And these are really hard experiments to get done, but they actually had human volunteers that came in for subsequent punch biopsies in their back skin, essentially. So you could actually trace these, they get samples from day 0, 1, 7, and 30, and did single-cell RNA sequencing. We analyzed independently their data sets, found the same phenomenon where we saw these P16 positive, P21 positive, K67 fibroblasts that peaked around day 7 and then went away. And they represented about 2% of that population. So everything's very consistent. And a similar ECM gene expression profile as well. So now we come back to the mouse and look to see what the differences are in aging. So in our 2-month-old mice, again, we see about 4% of the cells are these senescent fibroblasts, and about a 2.5-fold decrease in the aged mice, about 1.7%. These are mostly in the clusters of fibroblasts. We could actually stain P16 and PDGF receptor alpha, which is a marker of fibroblasts, and confirm that there was less of these cells in aging, which was quite surprising to us. When we next looked at what kind of SAS, again, this is the secretome of these cells, we found that the 24-month-old, the aged, had a much more inflammatory type of SAS that they were secreting into the environment, and also had a lot less of the ECM. So they're doing different things. And when we were able to look at the signaling output from these cells, what we found is that in young wounds, that senescent fibroblasts are broadcasting these signals that instruct the surrounding cells to properly build matrix, organize it. We have a lot of collagen, taste, C signaling, etc. For an aged wounds, this signal has been basically silenced, and you can't kind of guide the tissues towards wound healing and reconstruction. We've been confirming over the last couple of years that this happens in situ. So all the data I showed you was digesting the cells, the tissues, and doing analysis. And the new approach is now to actually look at the same kind of data, but in the tissues in situ. So we're using a platform based on MERFISH, and the idea here is that you have probes that can look at these individual gene transcripts in the tissues themselves. And with this particular technology, you can actually get subcellular resolution of where these transcripts are. And you get these beautiful images. This is actually some of our unwounded young mice skin. These are all just transcripts that are being detected in the tissue. If you take a deeper dive, you can actually see where they're actually localized. So we did these experiments kind of similar to what I showed you. Let me just orient you here because there's a lot of data on the slide. This is a young day 6 wound. This is an aged day 6 wound. These are classic kind of H&E stains. This is the epithelial surface on this young day 6 wound. This is the wound edge here. This is the actual wound itself. For the aged, it's kind of reversed. I apologize for that, but this is the kind of epithelium. The section kind of flipped over a little bit on itself, but this is the wound here itself. So with this technology, we can now use our triple marker and actually identify those cells that I just talked about and localize them to exactly where they are in these tissues. And I'm not showing the analysis here, but we find in aging that these cells tend to want to cluster. Whereas in the young wound healing, they tend to be a little bit more dispersed throughout the tissues, which probably has more of an impact if you think about the topography of where these cells actually are. And importantly that, we can actually detect what's surrounding these cells, so we can analyze the niche around them at any kind of level we want. I'm showing you here, each red dot is the senescent cell itself, and around we can actually characterize what cells these are actually interacting with. And if we look at the young wounds, they tend to interact more with endothelial cells and keratinocytes, whereas the aged tend to have less interaction with endothelial cells and interact actually more with immune cells. So it kind of reinforces the things that we're seeing here, that the young wound senescence response is really promoting every aspect of wound healing that allows it to continue, whereas the aged are kind of putting a big dampening signal on it. We reconfirmed kind of that SAS-related signaling and found, that there's more tissue-degrading pro-inflammatory secretome in the aged, more of a controlled matrix precision that was happening in the young. And on the right here, what you see is these K57, which is a marker of proliferation, to see what's happening around these niches. And it turns out you can see that there's very low proliferation around the senescent niches, whereas we still maintain the ability to proliferate around the young senescent niches, suggesting that they're not really doing the classic senescence things. They're really more instructing this to kind of continue. And we can see that this proliferation across different cell types that exist. So to summarize this first part, when you think about when you make that incision in your older patient, you're already operating on tissue that fails to mount the same coordinated biological response that makes wounds heal efficiently. Now, the senescent story is just a small piece of it. I get that. But when you think about it, you just got to think about the biology. There's something really happening that's preventing these wounds to heal at a normal pace. So in young wounds, these acute senescence are purposeful. We want to try to preserve this for coordination of healing. But in aging, this really corrupts its response. So maybe potentially this could be a target for removal, right? Because we have this aberrant response that doesn't seem to be facilitating repair. And I think when we think about the future is really can we incorporate technologies, omic technologies like spatial transcriptomics to think about how we can map where problems are happening in surgical problems, surgical issues. And you can think of many probably applications, certainly within the oncology field, of where we can use this type of phenotyping in making decisions. So I'm going to take a break and go away from the cell and actually talk about the matrix, because this is just as important as the cells themselves. So this is a project that we have going on with Jeroen Eijkmans, who's a biomedical engineer at Boston University. And what we're focusing on now is the matrix itself, because we know, senescent cells can produce matrix and they can alter the matrix. But what does that matrix actually do? If you produce a bad matrix, the thought is that the senescent matrix can actually reinforce senescence, and you have this feedback loop that basically reinforces this pathology. So the idea here is that the ECM just doesn't reflect senescence; it can actually perpetuate it. So they've built this really cool technology using 96-well plate format that they can synthesize these what we call 3D stromal microtissues. They have these pillars made of PDMS, and you can inject any kind of combination of cells or matrix, and these cells will reorganize the matrix to form these 3D microtissues you can see attached to the pillars here. And you can laser ablate, create these small wounds, and watch these microtissues heal over time. So you can manipulate the cell types, you can manipulate that matrix, you can test different drugs. It's a really fantastic model that they've developed. And so this is kind of what you see when we get these results. Let's see if I can get these movies. Sorry. What you'd see here is an ablation of this hole, and then you can basically see the cells migrate and repair this microtissue damage. But when we actually use senescent dermal fibroblasts, what you find is that the tissues are much less resilient to this injury. They can almost explode when you do this injury. And the microtissues that have senescent fibroblasts take forever to heal if they heal at all, suggesting that there's a lot of less resilience to injury and also impaired wound closure that happens in senescent dermal fibroblasts. Oh, here we go. Sorry. So again, here's that micro damage in the normal. You can see in the senescent really just kind of explodes here. You have less resilience, which you could kind of extrapolate to what happens in human age tissues. The other part of this is they can grow these cells and just isolate the matrix themselves. So you can grow normal fibroblasts in a plate and actually just decellularize that and grow and have the matrix itself. So this is normal dermal fibroblast matrix, and this is senescent matrix as well. And they can seed these microtissues with the same kind of constructs. And when you put the normal matrix in and you put normal fibroblasts in, they heal just fine. You can see the collagen type 1 is filling in this hole. But when you do the same thing and put a normal into it, a senescent matrix, they can't repair it as well. So they have a hard time actually repairing these tissues. And the matrix that they produce really is dysregulated and fragmented, something that you might see that occurs from aging. So to summarize this section, it's really Also, not just the cells themselves, but the matrix that leaves them behind that's also hostile to repair. So if you think about it, if that corrupted matrix still is there, then it can actually it can perpetuate even more senescence. So the next steps we're working on in this project is really to find the composition of what defines a young, healthy wound matrix versus age wound matrix, kind of at a proteomic level. And can we use that to inform how we can reverse engineer to get a younger matrix, basically promote healthier and more resilient tissues. So I'm going to switch gears again now and talk about more of a recent project that we've had going in the lab for the last year. And this comes with kind of the observation that we know, older patients tend to have worse outcomes after surgery, or if they have chronic wounds, they tend to have worse outcomes as well. And this gap continues to widen as this patient population ages. And we feel that the biology that's actually driving this is poorly understood at a mechanistic level and remains untargeted. So in both of these models of surgical wound stress or chronic wound stress, we want to ask the question is, what is the biology that makes older patients so vulnerable to this wound stress, and it's something that we could potentially intervene on. So I'm working with Lenora Thompson, who actually used to be a professor here at the University of Minnesota, and Vladimir Boskrov, who's a professor of dermatology at Boston University. And we tried to come up with a model that could help us explore what wound stress means and how it potentially can accelerate both functional and molecular aging and help us define what the mechanisms are. So we have cohorts of 5-month-old, 15-month, and 21-month-old mice that we get both males and females, and these are kind of your equivalent young, middle-aged, and aged adults in terms of human years. And in our model, we now put these mice through 3 consecutive wounds, put them through a lot of stress, wound stress. Over a 3-month period, they're always having to battle a wound and having to heal it over time. So we think this is a little bit more equivalent to potentially, a big surgery, a complication, a reoperation kind of thing in that sense, or potentially this idea of a chronic wound that always kind of cycles and never heals and then cuts back over multiple years. So we created this model. We did a lot of functional tests, which I'll go over in a second, and also harvested the tissues to see if we could find evidence of accelerated cellular and molecular aging that happens. And the data I'm going to share with you now is really just on the males today, because there are some pretty massive sex differences that we're finding. In general, our females cannot tolerate this kind of protocol. We actually have a lot of older females die through this process. We actually repeat some of the studies because their frailty and their tolerance for this kind of stress really was remarkably different from the males. So using this kind of repeated wound model, I'm just going to show you in these diagrams here. This is our 5-month, 15-month, and 21-month males. That first wound is shown in the orange and the second in the green. By the time the third wound happens in the 5-month, they do have some delays. And this becomes again more separated with aging. You can see now the first wound for the 15-month and the second wound are now separated, and the third one becomes even harder to heal. And by the time you get to 21-month, each one becomes sequentially harder to heal. So it really, aging kind of amplifies this loss of resilience that happens over time. So as I mentioned, we did a lot of these functional tests, put them on treadmills, make them do balance tests, etc. And what we find so far is that this kind of chronic wound stress really drives this progressive and age-dependent functional collapse. And if you look at activity, so this is kind of the most significant decrease that we saw. The animals that older animals that have undergone this wound stress really just stop wanting to move, and it's pretty remarkable. You kind of see this in the patients as well. Their voluntary activity just kind of drops to the floor. What you're seeing here are actually individual mice that we assessed before and after this wound stress. So each one is its own control, I would say. And what we found in the 15-month-olds that they actually dropped off to activity levels that were worse than a typical 21-month-old. So they really had a big deficit. And they also lost endurance on the treadmill. The only kind of motor change that we were seeing in the younger mice was the test for rotor rot, which is kind of a coordination-based testing. And we think this is probably due to some inflammatory deconditioning, which I'll explain in a second. So we took all these functional tests and created a kind of a composite functional score test for these and calculated the differences of what you would expect for a normal aging trajectory for that and then calculated how much aging was actually happening. And that's what I'm showing in the data here. So in our young kids, they basically gain about 4.5 additional years. Their behavior or functional capacity is like they're 4.5 years older than they actually should be. And as the aging went on, we actually find that in the age groups, that their functional performance is now a decade more than it should be after this chronic stress. So it's quite remarkable, and I think it's something that probably models what we actually see in our patients, especially the older patients that undergo these big surgeries. Next, we looked at some of the potential signaling mechanisms that are happening, and we looked at the serum cytokine profiles. So we wanted to see what cytokines were being altered during this chronic wound stress with the original hypothesis that this massive amount of stress would just lead to massive amounts of inflammation that couldn't be controlled. And what I'm showing you here, explaining, is looking at the full change of some of these serum cytokines in the mice that had received this chronic wound stress compared to mice of age-matched controls that have never had that stress before. Does that make sense? So anything above 1 means that it's higher in the wound stress cohorts. And in the blue is the younger cohorts, and in the kind of yellow is 15-month, and then the purple is the 21-month. And we saw this massive increase in inflammation across many cytokines in the young group, which I think may be driving some of those motor deficits that we're seeing. But remarkably, we see that in the 15-month and the 21-month, they actually start to have kind of immunoparalysis. We're not seeing that huge response in immune signaling that we'd suspected to see. And this actually parallels what you can find in sepsis, actually, when you have this initial sepsis response, you have this massive inflammation that comes. But over time, actually, this becomes immunoparalysis, and those organisms actually have diminished immune response and actually can have increased infection risk. So that's what we think is kind of happening in the aging space. I know it's a busy slide, but now we want to look at the tissues, what's happening to the tissues that is actually driving this process. So on the left side of the screen, we actually harvested the skin from this chronic wound stress model and analyzed the skin for markers of senescence and fibrosis, etc. And what you can see here in our 5-month, 15, and 20-month cohorts is basically gene expression for P16, which is that senescence marker. And the younger mice really didn't kind of accumulate these P16 positive markers in the skin, whereas in aging, they start to accumulate this damage, these senescent cells in that chronically wounded tissue, that it gets worse with aging. Surprisingly, we didn't find a lot with P21. We're still trying to answer what that question means. And the other finding is that we saw that there was increased collagen expression. And normally at this phase of wound healing in the remodeling phase, you really should be just remodeling. You shouldn't be having lots of expression of collagen. It's just reorganizing what's there. But so which happens in the younger mice. Then the age mice, they start to have this collagen expression that suggests there's kind of in this pro-fibrotic state that is abnormal. And we see evidence of MMP3, which is a major destructive metallic proteinase also when it's there in chronic states. So this is the bulk of expression in the whole skin. The next question I want to ask is, does this happen at the cellular level? So what we did is actually digested the skin from these samples, isolated and sorted for the fibroblasts themselves, and asked kind of the same question. And we submitted these for bulk RNA sequencing, and we saw the exact same trends. So this is bulk RNA data that's kind of showing you the same trends towards the younger mice having decreased P16, but the age mice really having this increase in P16, and really just recapitulated what we saw in the bulk tissue. So, the wound has completely healed by this time, but the biology is still remaining there. That's driving some of this difficulty in repair. I know this is a super busy slide. What I want to stress here is that what we see in aging, these are called volcano plots. So it's basically looking at the differential gene expression comparing two groups. And what we're comparing here is the skin fibroblasts from the chronic wound cohort versus age-matched controls. And anything in the red are actually individual genes that are really upregulated in this wound cohort, and in blue, they're below. This first column is showing you basically what happens in young mice, and you're seeing this massive change in gene transcription, right? It's pretty immense. This is a testament to more plasticity, more able to mount a regenerative response, which diminishes with aging. This is 15-month and 20-month. So that's one of the takeaway messages from this. The other important thing that we saw is we actually harvested skin from the ventral surface. So this is skin from the abdomen that has never seen a wound ever. It's never been touched by us. And these mice have only had that dorsal wound to see if there was systemic signaling that was happening throughout the entire body. What we found was similar, was that skin was changing in these ventral sites, suggesting that there's this chronic wound stress has systemic signaling that changes probably the landscape of the entire organism in particular. And finally, from this, I wanted to talk about aging. So we were able to use this new method called TACO, it's literally called TACO, or transcriptomic age calculator online from Gladyshev, who's a professor at Harvard. And what you can do is you can plug in your RNA-seq data into this online calculator, and it really allows you to actually look at transcriptomic aging to determine if these cells or whatever tissue you have is aged more or less than what you would expect. So this is the data from the dorsal skin here. You can see that the young actually have this kind of rejuvenated response, right? I just showed you that massive change in gene transcription, which suggests that there's some reprogramming going on, et cetera, that maybe brings these to a younger state. But what you can clearly see now in the 15-month and the 21-month is that they have this trajectory towards aging. So those skin fibroblasts that have been exposed to this chronic wound stress actually have a transcriptome that makes them a lot older than what they should be. And we actually see the same aging in the ventral skin. Again, skin that we've never touched is now becoming aged as well. And if you end up extrapolating kind of what these would expect, we're talking probably about a decade of aging in the skin fibroblasts from the 15-month and about a little less than a decade of aging in the ventral skin. So our next steps in this project over the next year is to find out what are these signals, what's being sent to actually drive wound stress to aging and accelerate aging in that manner. And if we can identify some of these features, then we can next design some therapeutics to potentially block what's happening. All right, so in summary, age wounds lack this correct acute wound cessation response for repair. So what does that mean? Your older patient that you're operating on cannot organize its own healing, and it's going to be delayed. Wound stress accelerates biological aging both locally and systemically. So any prior surgery, any wounds that this patient has, building of this aging phenotype to all the tissues that could potentially compromise any kind of future surgery that you have, including senescence and fibrosis. So, when that wound doesn't close, that it opens up, it's not always a technical failure, and you're battling some biology that has some meaning that potentially could be driving this. And finally, we can really have an opportunity now to measure this type of biology in our patients that we've never had before, which I think makes it really exciting. And the last part of my talk is a little bit more my pipe dream and kind of things I'm thinking about in the future. So this paper just came out 2 months ago from really the biggest leaders in the aging field to talk about this concept of geroscience to precision geromedicine. And the whole idea is that you could actually phenotype each one of us to figure out what are the pathways of aging that are being accelerated or suppressed, and can we design specific therapies that address each unique person's aging trajectory. And I think the big takeaway I had from this particular paper, and I know this is a big figure here, is all about the data that we can collect. There's a whole, there's a new concept of using this multi-omic data that we've never had before in terms of clocks, transcriptomics, proteomics, et cetera, that can bring a whole nother level of information to aging that we have never been able to do before. And kind of the goals of this are designing interventions that we can potentially use for geromedicine specifically, but I wanted to try to apply this to principles of surgery. So one of the interventions is being proactive, which means that we can potentially profile the aging landscape of the patient before you operate on them, either at the systemic level or at the tissue level. You can actually know the chronological age of that tissue before you actually go forward with surgery. And then you can prevent. So you can find out what the biological age issues are and potentially prevent that from happening with various interventions or target the aging biology behind it so you can intercept at any point of failure. And the idea for trials, I think, is perfect for surgery because we have these defined windows that we have We have a preoperative window. We know the surgical date. We know the actual surgery. We know the outcome. So we have this opportunity to really have points in time to work with that patient to optimize, measure, and see if we can have the outcomes that we want based on biology of aging. And it's happening already. So this is a study out of Denmark in 2025, published in Annals of Surgery, who used one of the methylation clocks called GrimAge preoperatively that they could use to predict how in pancreatic surgery, severe complications, statistical significance, failure to complete chemotherapy, and reduce survival based on basically taking a blood sample and predicting this. There was also this idea that we could have more defined biomarkers. This is a study out of the Mayo Clinic from 2020 that found 7 specific biomarkers in the blood plasma that can predict in cardiac valve and ovarian cancer surgical patients, ICU admission, adverse postoperative events, and rehospitalization. So just drawing the blood, looking at these particular factors, you could predict what was going to happen for these particular patients. So it brings us to this concept of senolytics in surgery. And I think this is the area that I really am trying to focus on in terms of how we can make significant impacts in aging biology, because we have a lot of the tools already. There are various ways to target senescence, including the senolytics, which induce apoptosis. You could target using CAR T therapy, actually, which is mostly in preclinical space. You can block the SASP using xenomorphics, such as things that are being secreted, and you can reprogram, which actually is not that far off, because there's reprogramming happening in humans right now in phase 1 trials for some retinal diseases. So this is not that far off, I think, in the future. So, do we target this pre-existing or baseline senescence that happens in aging and aged tissues, or do we target the senescence that arises in pathological states, or we do both? And how do we do this? Do we do it systemically? Do we do it locally? I think that this is already being kind of played out a little bit in the literature. This is examples of targeting senescence within transplant surgery. This is a paper from Stefan Tullius out of Brigham, a transplant surgeon there, who found that when you transplant organs from aged mice into young mice, you actually transfer the senescence to them and all the bad things that happen with it. But when they are able to actually clear all the senescent cells using D+Q prior to transplantation and then transplant those hearts to these mice, they found that there was a significant improvement in survival of both those mice and of the organs themselves. So, and this is a report from actually the group here in Minnesota that found in liver transplantation patients, if you looked at peripheral T cells P16 and P21 expression, you actually found a significant association with the patient's frailty and length of hospitalization, suggesting you can examine these things kind of in the preoperative space to create an aging signature for them. And this also happens in cardiac surgery. This is a recent clinical trial, randomized controlled trial that came out, I think published this last year, that used quercetin, which is part of the D+Q senolytic story. Gave these patients 2 days of quercetin before surgery and then through surgery for about a week. And what they found was lower discharge CRP levels. They found improved endothelial functions. They found decreased levels of senescence and inflammaging pathways. Postoperative atrial fibrillation was reduced with this treatment. But importantly, this actually only improved in males. They actually found the exact opposite in females, which is really important that we understand how these sex differences play out with regards to aging biology. So what are we doing in the lab here? This is the last kind of few slides here. We're trying to do a couple different approaches. So we first decided to try to clear senescent cells locally in the aged skin to see if we could impact repair capacity after that. So we have been using ABT-263, which is another senolytic called navitoclax. When we apply this topically to the skin of aged mice, we could reduce senescence levels by about 40 to 50%. And what we found was that this actually led to an increased level of wound healing after that. So removing these detrimental cells really seemed to restore the ability for these cells to repair. And we're now looking at the specific cellular targets for this, including we find that there's 3 clusters of fibroblasts that are sensitive to this treatment, as well as pericytes and some suprabasal keratinocytes. And the other work that we're doing is looking at D+Q at a systemic level, because we, in the literature or in the field, people are worried that this clearance might get rid of that beneficial wound senescence response, right? What we might find. So we focused on D plus Q and giving it systemically through what's called an oral gavage therapy. So starting on day 0 of the wounds, we actually systemically treat these mice with D plus Q. And what we found in the young mice is we didn't find any delays in wound healing. Thank goodness, right? So that you could, these patients that are on D plus Q may not have a problem with wound healing. When we did our age mice, we found that there was actually a statistically significant acceleration in healing with this systemic treatment. And what we think is happening is, like I mentioned before in the earlier part of the talk, there are differences, right? These senescent cells that show up in youth and aging have different phenotypes. And these drugs that we're using to target them really potentially target only one kind of subpopulation of those particular cells. So, in closing, I think the preoperative window is really the optimal therapeutic opportunity for making these interventions in older adults. And about 2 years ago, it took about 2 years, I helped build what's our geriatric pre-procedure clinic or perioperative care clinic with a geriatrician, Lisa Caruso, and a trauma surgeon, Sabrina Sanchez, who's interested in geriatric surgery. And we built this to be able to optimize care for older adults that are undergoing surgery, so they can come in, a couple weeks before their planned elective surgery. You get a full assessment by the geriatrician, and we can address things like nutrition and rehabilitation, pre-rehabilitation before they actually have that surgical procedure, to really optimize what their surgical outcome could be. But what I really hope we can provide in the future, and I don't think this is potentially that far off, is that we can actually start to measure biological age, tissue profiling, understanding what is the actual biological age of your tissues in your body before you actually move forward. I think that'd be an interesting data point for us, right? If you see someone that's 70 and their biological age is 95, how would that change how you're approaching your surgical procedure? And then we can start, as we start to define and measure aging in new ways, we can start to optimize how we treat that. Really, this will be a whole nother level of pre-optimization. It's actually targeting the mechanism of aging in these patients before we actually have the surgery. And the goal, obviously, would be to reduce the aging burden before you operate on someone, and also improve their, accelerate their recovery after. So my parting thoughts here are, the aging patient who will not heal is not just old. Their wound repair and senescence programs have been corrupted. Their tissues are biologically older than they appear. Wound stress does not reflect aging; it actually accelerates it. So prior surgeries, chronic wounds, etc., can accelerate biological aging, and your patient carries this with them to potentially get operate. Geroscience is changing medicine at a rapid rate, and I think our field surgeons have to play a critical role. The biology is becoming more clear, the tools are becoming more sophisticated, and we actually have this window that many other groups do not have to make these big differences in our patients. With that, I'll take any questions, and I really appreciate the time here. Last night we had a great dinner. Talked a little bit about what half the dinner was about World Cup, the other part was about aging. So it's fantastic that we were able to have that merging between surgery and the Aging Institute here, which was fantastic. So thanks so much. Happy to take any questions.
It's a great talk. Very interesting. So rehabilitation has been shown to have a lot of beneficial outcomes, especially for colorectal surgery and asymptomatic leaks. And I'm wondering if we have any data for what rehabilitation in the acute perioperative or preoperative setting does for senescence biomarkers.
Yeah, I think that's a great question. And just a caveat that it doesn't work for every surgery though, right? There's specific fields and specific patients where it works for, which I think is an interesting question itself. But why does it work? Rehabilitation work for some surgeries, some patients, but not all. But anyway, to answer your question, I think exercise is one of those widespread things that happen that can certainly impact senescence. No question. It impacts multiple hallmarks of aging, I think. So that component of rehabilitation, at least in the exercise space, certainly can impact senescence. For sure. I don't think people have really measured it at that level, but at least not humans.
Thank you for the talk. Are you also thinking, you mentioned working with Dr. Sanchez, are you also looking at trauma as a potential insult as well, and what that does to?
Yeah, the literature actually shows that something like trauma and these acute can actually have a temporary increase in biological age, at least by epigenetic clocks measurements. It's a little bit tricky because some of the when you measure these clocks like a day after trauma or a week after surgery, these are peripheral blood tests, right? And what happens in trauma? What happens in the acute space? Your blood cell, everything's different, so it's kind of hard to extrapolate exactly what that means. That being said, I think the literature shows that surgery can induce this transient epigenetic aging that happens that resolves. I think it probably has more to do with the blood that's changing over time. But it's Sabrina Sanchez is actually interested in this from not necessarily a biological standpoint, more of a patient care standpoint. And she's developed these frailty pathways that we use in our hospital now for patients that immediately when a patient is flagged frailty, a whole order set comes up and you have to kind of implement different ways. So she's kind of tackling it from a different perspective, though.
So great talk. Over time, exercise and weight loss. They change senescence.
I think the exercise story is there. Weight loss, I don't know, to be honest. I don't know if we're all. Yeah. When you think about our patient population, right, the older patient, how many of them are going to start exercising? Right? If there's other things that we can figure out that target that, maybe that's another group. Not to say that they can't, but sometimes it's more challenging in the older adult population. Thank you. I know that was very heavy on the data, and I appreciate you all. I stayed awake. I think at least had 90% of you were eyes still open, so I appreciate your participation in that. So thanks so much.
I think that they're at school now. I'm already done. I think it's
This one was lower.
I'm like sitting
No, kind of like things. It's like weird to things, but let's make sure.
and then went to the
My question is, why didn't you guys get kidding?